Shane slammed the door of the tractor shut and hurried as quickly as he could back inside. It was a bitingly cold November morning, and he'd be happy to get inside the central heating and the warm air once again. Winter was creeping up faster than he'd liked this year. He switched on the radio as he made some coffee and toast, but it was just coverage of the election results. Goddamn Bill Clinton gloating and giving his slick speeches. But Shane knew this just meant more of the money he made with his sweat and blood was going to be funneled to the government for boondoggles and to line the pockets of elites somewhere thousands of miles away on the other side of the country. Here in Idaho, there was a potato season to plan, and he had a tough decision to make. His farm wasn't huge, just a few hundred acres, but still the seed potatoes were probably his biggest cost each year, easily over $100,000. Mistakes could cost him and his family their house, their whole lives really. More and more of his friends and colleagues in the potato industry have been switching over to this new product from Monsanto. Was it time to make the switch? These new potatoes might make all the difference. Pesticides were getting less and less effective against the potato beetles that kept there munching through his livelihood every year. And if these potatoes were as resistant as they say, it might be worth a sticker price. But there were all these forms he'd have to sign, made him nervous. What exactly was he signing away? He'd done his research. He'd have to set aside a decent chunk of his field as a refuge for non-GMO potatoes so that the bugs didn't get resistance to the thing. What was it? BT, that's right, that they put inside the potatoes. That would complicate the planting. The complexity cost money. He took a few more sips of his coffee and went about his day. He placed a call to his Monsanto distributor, Rob. Nice guy. They had a little chat, and Rob was able to give him a good enough discount to get him to yes. When the 1997 harvest came in, Shane was up 5% on the year before. More than enough to make up for the more expensive seed potatoes, and he couldn't be happier with his decision. Shane was not alone in his calculations and in the returns that he saw. The NewLeaf potato from Monsanto was the first of a new kind of GMO crop product, resistant to insect pests thanks to a transgene taken right out of a soil-dwelling bacterium. The uptake was strong, and farmers were generally pleased with the product. And yet by 2001, it had disappeared from the market. What happened? Let's find out. Welcome to Modified. I'm your host, Dr. Orlando DeLange, ex-plant scientist and current science teacher. This podcast is my attempt to guide you through the science of GMOs and hopefully just enough of the history and the social context to give you a path forward in your own thinking. And if all else fails, I hope you'll enjoy some of the surprising stories and fascinating facts in the world of genetically modified plants. This is episode 12, BT Crops, GMOs Go Mainstream. I was originally planning this as an episode of two halves. I would start with BT and I'd move on to Roundup Ready crops. Each would get about 10 to 15 minutes, just giving you a bit of an overview. But it was just too hard to hold back on what I think are just the surface level facts, with only a little contextualization. So I'm giving each their own episode. So episode 12, this one is all about BT crops, and episode 13 will be about Roundup Ready crops. And then after that, episode 14 will be our second and final installment of this season of Your Questions Answered. So send in your questions to orlando@modifiedpod.com and I'll do my best job to answer them. But without further ado, let's dive into BT. Nazis are roaming the streets, looking for anyone who seems to fit a rough profile of undesirable person. They regularly detain people, rough them up, and many never return. It's 1944, and Ernst Berliner and his wife are among the lucky few. After a brief detention, they are released. The Gestapo have been watching Dr. Berliner for years, convinced that he must be Jewish, despite what he may tell them that his family long since converted. Like the majority of Germans during this time, he just wanted to keep his head down and stay out of politics. And plus, he was too useful to the Nazi state. He was by now fairly senior in his career, where he traveled Europe, training technicians in the scientific practices of crop protection. When he was just a lab assistant, back in 1911, he made his biggest discovery. If he were alive today, he'd be shocked at just how big of an impact that discovery made, and how much it would go on to drive a wedge between agricultural scientists and so many other groups within society. But really, living in mid-20th century Germany, he had bigger things to worry about. What Dr. Berliner had discovered earlier in his career was a pathogen, a bacterium to be more specific, one that had sickened a whole shipment of moths. Specifically, these were flower moths. That's flour, the cereal product. This was a problem in the near term. They needed the moths alive for their research. But what if this pathogen could be used as a weapon against these annoying and economically devastating pests? He named the bacterium Bacillus thuringiensis. Like so often when it comes to scientific names, particularly those that are a little bit longer, a bit of a mouthful to pronounce, the full name is shortened to just the initials, BT. It took until the 1960s till it became clear that what's happening is that these bacteria produce a specific toxic protein packaged into crystals that surround their spores. The exact molecular details of this process are still being researched by labs around the world, but the key steps are known. The toxic protein, known as a cry protein, C-R-Y, cry, is eaten by an insect larva. Specifically, the one that was discovered by Dr. Berliner works only on the larvae of moths and butterflies, the taxonomic grouping Lepidoptera. These larvae are sometimes known as caterpillars, but particularly the ones that are pale in color are known as worms, like the flower worm mentioned earlier, which is the larva of the flower moth. And importantly for the world of GMOs, they also include bollworm, which is a major pest of cotton, corn, and soybeans. Inside the gut of the larva, the cry protein latches onto a specific receptor in the membrane of gut epithelial cells. That means the cells that are the outer lining of the gut. I want to emphasize that the protein will only attach to a specific corresponding receptor protein in the insect gut. That's important when thinking about whether or not we should be worried about unwanted impacts of Bt crops on other insects. Once the cry protein has found its receptor, it changes shape and links up with other cry proteins to essentially punch a hole in the cell membrane. This allows ions to flood out of the cell and kills the cell. That in itself is pretty bad, since without functioning gut cells, the worm cannot eat. But even worse, the dying cells create gaps in the lining of the gut. And that basically allows an entry point into the blood underneath. That is very bad news for the moth, or for the larva. For humans and insects alike, our epithelia, that is our barrier tissues like skin, but also our gut lining, are keeping us alive every day and night by acting as a barrier against the vast majority of pathogens, keeping them away from our much more sensitive tissues. And it is very catastrophic if pathogens get into the blood, where they can quickly circulate around the body. In humans, that would trigger a massive immune reaction called sepsis that can be quickly fatal if not treated. Insects work a little differently, but similarly, when pathogens get into the blood, it's very bad news for them. And that's what the cry proteins allow by punching holes in gut cells, killing the cells and allowing other pathogens to then pour through the lining of the gut. So, in end effect, cry proteins, often just called Bt proteins or Bt toxin, are fatal to the insect larvae that they target. Even though the molecular details are a more recent discovery, the insecticidal action of these proteins and the bacteria that produce them has been well known by agriculturalists since not long after Dr. Berliner first described them. And indeed, just about the same time as the Nazi secret police force were hassling Dr. Berliner, so that's 1944, a French company started to sell a product made from this bacteria. The product was called sporine, and it was only available briefly. It was a powder to coat crops with. It was 10% Bt bacterial spores mixed in with some flour, perhaps to make the powder attractive to flower-eating insect larvae, or maybe it just helped the powder stick to the leaves. It was hard to find much information about that. But while Dr. Berliner survived the Nazi period, sporine did not. Production was halted as France was invaded and occupied. However, in the 1950s and 60s, the same idea was tried again, with commercial products developed and offered independently in the USA, USSR, and China. I don't know how widely these products were adopted, but suffice to say, Bt bacteria were well known to farmers and agriculturalists by the 1980s, and so it shouldn't be surprising that Professor Mark von Montagu in Ghent, Belgium, directed his team of genetic engineers to synthesize the gene encoding cry protein and insert it into a plasmid, and then use Agrobacterium tumefaciens to deliver that gene into the genome of tobacco cells. You may remember Professor von Montagu from episode 10. He was the one who in turn inspired Dr. Luis Herrera Estrella, who went on to work with him, and they together demonstrated the first use of agrobacterium to make a transgenic plant. The Von Montagu lab didn't give up their work on developing and promoting this new technology, and they saw the cryotoxins of Bt bacteria as an excellent application. They were not wrong. They published their work in leading science journal Nature in 1987, showing clearly that the transgenic tobacco plants they created in their lab were totally protected against the tobacco hawk moth larvae experimentally introduced into their greenhouse. The larvae simply shriveled and died when they tried to eat the plant. Now, what you should picture happening in the 1980s and continuing to the present day is not that there are two totally parallel worlds of academic research labs and then big agritech companies like Monsanto. Instead, there is a lot of interaction, and mostly this is one way from academia to the companies, at least in terms of the flow of ideas and technology. All academic research has to be published, that's kind of the point, and it also gets presented at conferences and shared at smaller meetings, often long before publication. Plus, the people who get their training in academic labs go on to work at the bigger companies after they finish their PhD. So there's a continuous flow of information and people. And because of that, it's not surprising that Monsanto essentially developed their Bt transgenic crops at roughly the same time as the Von Montagu lab were publishing. Just as in the same way as Calgene developed their Flavor Saver tomato at the same time as Professor Grierson's lab in the UK developed their own polygalacturinose RNA silenced tomatoes. To this day, research in academic labs is followed closely by private companies, or private companies are directly spawned by research labs. And this isn't predatory, it's the system acting as designed. Basically, most funding for plant science research is provided with the intention that this should feed back into agriculture. The idea that science is there to yield useful technologies is the basic premise that drove huge investment in scientific research over the 20th century, particularly in the USA, but certainly not only there. The USSR also, during the Cold War, invested hugely in science, not out of a particular passion for the pursuit of knowledge, but because of what it can do. Anyway, that's all pretty obvious, I'm sure, but what that leads to in practice is a deliberately blurred line between academic research and private enterprise, one that, if anything, has been encouraged to become much more blurred over the last few decades. Up until the 1980s, for example, it was taboo for an academic scientist to directly involve themselves in private enterprise while retaining their academic position, for example, setting up a company directly based on their own research. That is now commonplace and, in fact, positively encouraged. It seems that through the 1980s, Monsanto's research teams were working on a lot of the same ideas that researchers in academic labs were working on. And the only thing I know is it's not a coincidence. But it could be they were just keeping up to date with the literature, following up on all the ideas being shared in publicly available peer-reviewed research papers. Academic scientists tend to be under intense pressure to publish a lot of these research papers, so a project that may take many years will basically be described in installments, with additions every few years as the research develops. And research isn't just shared via journal articles, as I mentioned earlier, it's shared at conferences that scientists from private companies are generally welcome or encouraged to attend. And like I mentioned, labs are not one person, they are teams, and most of the members of the team in an academic research lab are only there for a few years and they move on. And if it's a plant science lab, they very often move on to work in the agritech industry. The world's first commercial Bt crop mirrors the story of the Flavor Saver tomato. A burst onto the market with a decent amount of enthusiasm and then a rapid fall after just a few years on the market. So for Bt, that crop was the NewLeaf potato, and it lasted 6 years, slightly better than the Flavor Saver, from 1995 to 2001. And I will share a little bit about the NewLeaf and what caused its downfall in just a minute. But unlike the Flavor Saver, while the NewLeaf potato may have fallen pretty swiftly out of favor, the basic concept behind it of Bt crops have been a long-term unmitigated success from the perspective of those making and selling them. Bt, or more accurately, genes encoding cryproteins from the bacterium Bacillus thuringiensis, has been introduced into a wide range of crops, and most notably into corn and cotton. In the US, the percentage of total corn and cotton plants that contain Bt transgenes grew pretty rapidly to achieve 20% of total planted acres in their first few years on the market, that is, the late 1990s. But from there they grew even faster and further over the first decade of the 21st century, currently reaching roughly 90% of total planted acres by 2014, where they have stayed ever since, according to the latest USDA data linked in the show notes. So to put that another way, 9 out of 10 corn and cotton plants growing in the USA have a transgene in their genomes, allowing them to produce cry proteins. A recurring theme for Season 2 of this podcast will be the powerful and successful resistance to GMO crops in Europe in the 1990s, which remains the majority social position. But don't think that GMOs are restricted to just the USA. Similarly, Europe is not isolated in its opposition. It's much more of a global patchwork. In the Americas, Brazil, Argentina, and Canada are among the other countries besides the USA where Bt crops and the majority of acres planted, at least for those crops where such cultivars are available, such as corn and cotton. Whereas in other countries, notably Mexico, GMO crop cultivation is forbidden by law. In Asia, there are several countries, including China, which have a lot of GMO acreage, although not the majority. In India, most cotton planted is Bt cotton, and that is a lot of cotton given that India produces a quarter of the global supply. I am planning to devote a whole episode in Season 2 of this podcast to Bt cotton in India, because it is one of the most researched case studies into GMOs and their costs and benefits, both economically and socially. But one thing that makes it so interesting is that most cotton in India is GMO, specifically carrying a cryotoxin transgene, but since 2010, there has been a moratorium on commercial cultivation of GMO food crops in the country. Africa has, over the last few decades, been something of a battleground, with pro- and anti-GMO forces battling to win over governments, resulting in another patchwork situation. South Africa was an early and enthusiastic adopter, while many other countries have outright bans. Kenya is just one example of the sort of muddy middle ground that exists in many countries in Africa. Kenya had a strict ban that included not just cultivation, but imports of GMO crops until 2022, but seems to now be heading towards widespread cultivation of Bt corn and cotton, among other GMO varieties. And in Europe, which as I mentioned has been a global center of anti-GMO sentiment, GMO crop cultivation is banned in most, but not all countries. But even those bans do not mean that importation is banned, just growing the crops, even if there is some additional bureaucracy involved. Within the EU, Spain is an outlier for having a significant GMO acreage, though even then it's pretty small potatoes relative to somewhere like the USA. There is just one GMO crop planted in Spain, a GMO corn cultivar, and it's actually a diminishing percentage of total corn crop in Spain. That one variety is called MON 180. It is specifically a Bt corn variety, and it was developed in the mid-1990s by, perhaps unsurprisingly, Monsanto. And so let's use that reminder to go back and check in on that first ever Bt crop, the NewLeaf potato produced by Monsanto. One twist I've left out till now is that the NewLeaf actually had 3 transgenes. One was just a gene used in selecting transformants, if you remember all the way back in episode 10. It's really important to have that sort of thing to find the successfully transformed Cali within the sea of little green blobs during the plant transformation process. But the other 2 were transgenes that have some protective effect for the crop. One is the gene for Cry3A, a cryotoxin protein, and I'll get to that in a second. The other was PVYCP. That stands for Potato Virus Y Coat Protein. I'm not going to go into a lot of detail on this, but next season I plan on doing an episode about GMO papayas in Hawaii, where a similar strategy was used. But basically, by inserting a gene from Potato Virus Y into the genome of the potato itself, the Monsanto scientists made the potatoes resistant to the virus. And this worked in a similar way to the way that inserting a reverse mirror image version of the polygalacturinose gene into tomatoes reduced expression of that corresponding gene in the tomato. In this case, the viral gene in the potato essentially primes the RNA silencing machinery of the potato to find and destroy viral RNAs when they are detected. The actual molecular machinery involved is different, but in terms of end effect, you can think of this like a vaccine against potato virus Y, but one that is built into the genome. it renders the potatoes immune to the virus through a system that recognizes and destroys the virus if it invades. Potato virus Y is one of many viruses that plague potato farmers, and yes, plants very much suffer from viruses. Viruses are really amazing. They're basically DNA that has become independent and self-replicating, and they demonstrate a level of efficiency that feels both beautiful and terrifying. Viruses are not like other organisms, and there's an ongoing debate whether they should even be considered living organisms at all. And their unique properties make them particularly devastating as pathogens, as well as offering some unique and powerful ways to protect against them. That is true for viruses that infect crop plants as much as it is for those that infect humans. Now back to that cry protein. The cry3A gene, just to restate again for clarity, is what we'd call the Bt gene. Cry3A is a gene from a particular strain of Bacillus thuringiensis. Oh my goodness, now I can't even pronounce it. Bacillus thuringiensis, or Bt bacteria. And at this point, I should point out an important feature of Bt transgenes that I've only hinted at so far. There are many, many different versions. So there are many different Bt genes, or more accurately, genes encoding cry proteins. They all work in a very similar way, but they differ at a number of positions in the protein they encode that allows each cry protein variant to match with a different target receptor protein in the gut cells of the insect that they have evolved to infect and ultimately kill. So each cry protein has a different specificity in terms of the insects it can kill. And that is great news for companies like Monsanto, but not restricted to Monsanto, which create Bt-based GMO crop varieties. Whatever particular insect pest is threatening the crop of interest, you can probably find a naturally occurring cry protein variant out there that will kill that insect, and even more importantly, only that insect. If you recall from earlier in the episode, cry proteins were first discovered and described in the context of moth larvae. Those are what we call lepidopterans. That's one of the major groups of insects. I'll try to refrain from going on a long rant here, but insects are just some of the most amazing organisms ever to evolve, and it's fair to think of them as the other major lineage of animals that took over and dominated the land of our planet. They actually made it onto land before the vertebrates, which is the lineage that we're part of, and so they've had a long time to diversify into a lot of different orders and families. These numbers change the more we learn about biodiversity on Earth, but insects overall represent the majority of all known animal species. They are crazily diverse. So anyway, back to the NewLeaf. The GMO crop wasn't designed to protect against moth larvae, but from the larval and adult Colorado potato beetle. Beetles are a totally different order of insects, the Coleoptera, and of the roughly 30 orders, that's the major groupings of insects, they are the most diverse, with almost 400,000 known species of beetle. And this beetle, the Colorado potato beetle, is a major problem for potato farmers, and therefore for the rest of us who love to eat potatoes. The humble-looking potato achieved global superstar status in just a few hundred years after Spanish colonization of the Andes led to its export across global trade networks. And by now it is deeply embedded in cuisines from Ireland to India and from Kenya to Kansas. The NewLeaf was well-timed because by the mid-1990s, the chemical pesticides that farmers had been relying on for decades were failing as the insects evolved resistance to them. I think it's worth pointing out that chemical insecticides were only required because 1. American agriculture is based around huge monoculture fields that are veritable banquets for pests and pathogens and provide ideal environments for the evolution of new pest and pathogen strains adapted for the diet in front of them. And 2. American consumers demand abundant and cheap food, so anything that lowers yields will end up devastating farmers because they live on very thin profit margins. There are reasons why both of these features of American agriculture developed, and why they have been exported around the world to become common to most agriculture. For now, I'll just throw it in as a reminder that when we're thinking about GMOs, it's important to remember that they exist in a broader context of science, technology, economics, and political decisions that are all interconnected. Anyway, on face value, the NewLeaf seems great. It helps out farmers by keeping pests at bay, without the need to spray a bunch of pesticide. And it's anyway more effective than the available pesticides. So why did it ultimately fail? Well, it's complex, but three key regions emerge in what's written around it: consumer resistance, complex requirements for farmers, and new pesticides coming onto the market. Starting with the last one, basically the problem the NewLeaf was addressing, the lack of efficacious pesticides, was solved in the late 90s with the launch of imidacloprid, I'm not actually sure how to pronounce that, from Monsanto rival and now owner Bayer. Imidacloprid worked really well on the potato beetle and the aphids that transmit potato virus Y. And given the other issues facing the GMO potato, this really took away a major incentive from growers. You probably haven't heard of imidacloprid, obviously I hadn't, if you don't work in agriculture, but you might have heard of a class of pesticides they belong to, neonicotinoids. And that's because they are in fact now restricted, though not banned in Europe and in the US, because decades of research showed that they harm wild pollinator populations, including bees, contributing to the huge declines in bee populations after the last few decades. So neonicotinoids have gotten a lot of press coverage over the last decade or so. So maybe the bees would have been happier if the NewLeaf had been more of a success and people hadn't switched to using imidacloprid, I don't know. Anyway, another problem was that the NewLeaf was not just expensive, but required farmers to set aside 20% of their field for non-GMO planting in order to reduce the risk of the potatoes becoming resistant to the cry-free-A protein inside the GMO potato plants. Just like with antibiotic-resistant and human pathogens, overexposure increases the selective advantage for those bugs or beetles that have rare mutations allowing them to evade the antibiotic, or in this case, the killer protein lurking inside the plant leaves. By having non-GMO refuges in the field, these rare beetles that happen to be resistant don't breed and spread so rapidly because their selective advantage is only relevant in the GMO portion in the fields. Okay, seems sensible, but that means the farmer is paying a premium for a product they can only use on 80% of their acreage. The rest has to be planted with non-GMO potatoes, and presumably those are going to get normal insecticides. So now it's a complex situation where you need all your gear and chemicals anyway to deal with just 20% of your acreage, and then you need to track what got planted where and make sure it all gets treated appropriately, and that all costs time and money, not a winning proposition. But the number one reason it failed, and why to this day GMO potatoes exist but are not dominant in the market, was concern and resistance from consumers. We'll explore much more next season how that concern and resistance was raised and amplified in organized ways. But ultimately, if consumers don't want to buy the potatoes, farmers don't want to grow them. In particular, by the late 1990s, the early years of the NewLeaf potato, the public resistance was getting really strong and really present in media. So much so that McDonald's was forced to take a position and state clearly that they would not use GMO potatoes in their fries. And that almost single-handedly sealed the death sentence of the NewLeaf potato. Well, I mean, maybe it did, maybe it didn't. But it's at least a very clear indicator of the public mood that let farmers know that planting these potatoes was a risk. The other more successful markets for GMOs, corn, cotton, soybean, those are all crops that do not make it to a consumer intact in most cases. Yes, you can buy whole corn cobs or whole soybeans, aka edamame, but the vast majority of the harvest of corn and soybean is processed into products that you won't notice or even ever interact with. In the case of corn and soybean, that's because you won't be the one eating them. Most soybeans in the US and a good chunk of the corn is sent to be processed for animal feed. And another good chunk of US corn goes to make ethanol to get blended with petroleum to go into car engines. And then cotton, well, that's never been a food crop. So a GMO potato or tomato, for that matter, is a very different prospect. Those crops are grown mostly for human consumption, and even in processed forms, you generally know that you're eating a potato or a tomato. And so you're much more likely to care about whether it is GMO or not. Today, there are about 400 million acres every year that get planted with BT crops. That's roughly one Alaska's worth of BT crops. According to a 2023 paper that I'll link in the show notes, that is. And those acres are overwhelmingly corn, soybean, and cotton with a little sugarcane. I won't go into it too much in this episode, but I'll just emphasize briefly that while Monsanto was the first to bring a Bt crop to market, they are not the only ones. As explored in the episode, Bt bacteria have a long history as a natural insecticide, and so there are many different Bt strains with different cryprotein genes, and so no one company can own the patent to all of them. And yes, of course, patents are going to be something I'll explore more next season, and they'll come up a fair amount in our episode 13, all about Roundup Ready crops. But anyway, Monsanto is joined by other big players like Syngenta, Pioneer, and also government research facilities, which have been particularly important in developing Bt crops in China and India. So by now, there are tens, if not hundreds, of different Bt crops grown around the world, all of them different varieties of corn, soybean, cotton, or sugarcane with different cryprotein genes, sometimes multiple different cryprotein genes stacked into one genome to provide better protection against the insect pests that might otherwise develop resistance. Like any conventional insecticide, and just like the antibiotics we use, resistance to cryproteins is an issue. I'll try not to give a whole evolutionary genetics lecture here, but briefly, when I say develop resistance, this isn't something that happens in the lifespan of one insect. Instead, remember, insect pests tend to advance through fields as a large population, thousands or millions of individuals. And within that population, there will, by pure chance, be some DNA mutations, and that will, for example, sometimes be a mutation in the gut protein that is the receptor for the cryprotein. The mutation might slightly alter the shape of that protein, such that the cryprotein basically can't find it and latch onto it anymore. The insect with that mutation will be able to survive, feeding off the plants that contain the cryprotein, and that means that it'll be able to have a bunch of babies, and those babies will inherit that DNA mutation and pass it on, and so on and so forth, until you have whole populations of insects resistant to the cryprotein. And that's, again, pretty much the same way when we talk about bacteria developing resistance. To antibiotics, that's what's happening. And this process can actually happen pretty fast because of what we call the strength of the selection pressure. The selection pressure is the environmental condition that the DNA mutation exists under, and that determines whether individuals with the mutation are more or less likely to breed and pass on their genes. In this case, the pressure is as strong as can be, because individuals without the mutation will die. The appearance of a mutation that confers resistance is random chance, and sometimes by random chance it doesn't happen for years or years. But once it does, resistance can start to become the mainstream for that insect species in a given region of the world within the span of a decade. That is really part of the constant fight between agricultural technologists and the pests and diseases trying to eat the crops. A fight that has been made particularly bitter in the last century or so as large monoculture fields appeared, a perfect evolutionary environment for resistance to evolve. Anyway, when it comes to Bt, there has been and will continue to be instances of resistance to particular cryproteins appearing in particular insect species in certain regions. That means in some regions where a particular insect pest develops resistance to a particular cryprotein in a Bt crop, it will render that particular crop line ineffective. But that overall, most cryproteins continue to be effective even after decades of use, and even where resistance does develop, it tends to be limited to a specific part of the world. One reason that resistance hasn't been more of a problem for Bt crops is actually the refuge areas mentioned earlier. Keeping some of the field planted with non-Bt crops reduces the intensity of that selection pressure, and which slows down the spread of those resistance mutations, or to use the technical term, resistance alleles, when they do appear. Despite the problem of resistance developing in some insect populations, Bt crops continue to be popular and widely planted, and most varieties remain effective, and new varieties continue to be created. So overall, Bt crops have been one of the major success stories when it comes to GMOs. Before we close out, I do want to touch on the issue of Bt crops harming non-target insects. This has caused a lot of concern, and I think it's a pretty reasonable concern. I might well do a whole episode next season on Silent Spring and the growth of the environmental movement since the 1960s. But at any rate, there are good reasons to worry that agricultural technologies promising to eradicate insects that kill crops might just eradicate pretty much everything they come into contact with, and most notably other insects, the ones we definitely don't want to kill because they are crucial to ecosystem health. Also because they are beautiful creatures deserving dignity and respect, at least in my opinion. These concerns seem to be scientifically supported when a paper came out in 1999 showing that pollen from Bt corn could kill monarch butterfly caterpillars if it is dusted onto their favorite food, milkweed plants. However, a spate of follow-up studies show that this wasn't true at more realistic levels of pollen, and indeed real-world analysis has not shown any effect of planting Bt crops on the populations of monarch butterflies in those areas. And the same has been found for bees. For bees, it's maybe not that surprising in that bees come from another order of insects, the Hymenoptera, that is very different from the ones that tend to be targeted by cryproteins. The cryproteins used in crops actually grown widely do not target Hymenoptera, and cryproteins are known to be very specific, at least within their taxonomic group of interest, like their order that they target. Since many commonly used cryproteins target moth larvae, aka caterpillars or worms, it's not surprising that you might have a situation where other caterpillars were killed as an undesired side effect, but bees is a bit of a stretch. Still, the data do not seem to support major impacts of Bt crops on any sort of other non-target insect populations. I'm going to link some articles in the show notes if you want to read more about that. While it is, I think, a really good idea to scrutinize the broader ecological impacts of any agricultural technology, Bt crops do not seem to harm insect populations beyond the specific pests they're designed to target. And so, as we finish up, what can we learn from the story of Bt crops? In a way, they are the ideal transgenic crop. The NewLeaf potato was a disappointing start, but not really representative. They are ideal because it's a situation where a single gene, in this case the cryprotein gene, confers a powerful, desirable trait that helps farmers cut costs by not having to buy pesticides, and so they get higher yields, and that in turn contributes to an agricultural economy that allows consumers to benefit from relatively cheap produce and other agricultural products like cotton. And since the Bt crops on the market aren't ones that we tend to be directly eating in any recognizable form, the average consumer doesn't even have to think too much about it. Those factors together are what I think make Bt crops seemingly so ideal, though as we'll explore more in Season 2, that appraisal can be and should be contested. In our next episode, we'll look at a type of GM crop that has long been much more controversial, herbicide-tolerant GM crops, or as they're better known by the original brand name, Roundup Ready. But for now, we'll end with our 5 take-home messages. Number 1, Bt stands for Bacillus thuringiensis, a bacterium that naturally carry cry genes in their genomes that encode proteins that can kill specific target insects. Number 2, Bt bacteria have been directly applied to crops for many decades as a pesticide. Number 3, cry genes have been introduced directly into crop genomes since the mid-1990s, and this remains commonplace and widespread, specifically for cotton, corn, and soybeans. These crop varieties are referred to as Bt crops. Number 4. The first Bt crop variety was released by Monsanto in 1995. It was the NewLeaf potato, but it was only available for 6 years. No Bt potato varieties are currently sold anywhere. Number 5. Since the first variety in 1995, hundreds of different Bt crop varieties have been grown and sold, only some of those produced by Monsanto. I realize I've been negligent in the last few episodes on calling out a few particular sources I found helpful in writing the episode. So I want to get back to that, and I want to acknowledge the chronicle of the ups and downs that made Bacillus thuringiensis a natural insecticide by Ninfa Rosas Garcia in 2023, and I want to acknowledge Global Patterns of Insect Resistance to Transgenic Bt Crops, the first 25 years, by Tabashnik, Fabric, and Carrier, also 2023, and I'll link those in the show notes along with other sources. Our next episode will be about Roundup Ready crops, but as I mentioned at the beginning, after that, episode 14 will be a second and final Your Questions Answer episode for this season, and I'd like to solicit any questions you have about GMOs and GMO crops. So please send them in to orlando@modifiedpod.com. That's orlando@modified.com, and I'll look forward to answering them. Bye!